A man is alone in a stone room in northern Italy in October of 1978, holding a camera he has used 1,000 times before. Except this time, his hands will not stop shaking. He has photographed factories. He has photographed medical procedures. He has spent his career capturing things exactly as they are, without embellishment, without belief, without agenda. He is not a religious man. He did not come to Turin looking for God. He came to document a piece of cloth, 5 days. That is all the time his team has been given. 5 days of unrestricted access to an object that has been locked behind glass, wrapped in silence and legend for 400 years. And somewhere in the middle of those 5 days, staring through his viewfinder at a faint, sepia-colored image of a crucified man, this skeptical, methodical photographer starts noticing things that do not add up, things that will not leave him alone for the rest of his life. His name is Barrie Schwortz. In 1978, he was 32 years old. He had no way of knowing that this 5-day assignment would become the defining work of the next 5 decades of his life, that he would still be investigating this same question at 78, that new technology would keep arriving, decade after decade, promising to finally answer it, and that each new answer would somehow only deepen the mystery. This is a story about one of those moments. It begins in 1978. It reaches its most important turn in 2026, when scientists finally had tools capable of reading something that had been sitting invisible inside the cloth's fibers the entire time. Biological traces so faint, so degraded, that no technology before this could have found them at all. And when they did find them, the results did not hand anyone the clean answer they were hoping for. They handed them something far stranger, a tangle of human lives, layered on top of each other across centuries, hidden inside threads of linen. A biological record so complicated that it raises a question nobody fully expected to be asking in 2026. After everything modern science can now do, is it even possible anymore to find the one signal out of all of them that actually matters? Let's go back to where this started. Because to understand why the 2026 discovery hit as hard as it did, you have to understand exactly what came before it, and exactly what kind of man was standing in that room in 1978, watching something happen that he could not yet explain. Barrie Schwortz was, by his own long public account, an unlikely candidate for this role. He was Jewish, with no particular religious investment in Christian relics, recruited not for his faith, but for his technical skill. When the offer came in 1977, joined a team of American scientists traveling to Turin to examine the shroud, his first instinct was closer to indifference than curiosity. A medieval painting, probably, a tourist relic, an interesting photography challenge, and nothing more. He said yes anyway. And in October of 1978, he became the official documenting photographer for a team that would come to be known as STURP, the Shroud of Turin research project. Physicists from Los Alamos, chemists, forensic specialists, over 30 scientists in total granted something that had never happened before and has never happened again since: direct, hands-on, round-the-clock access to the cloth itself. 5 days, October 8th through the 13th. Scientists sleeping in shifts while others kept working through the night. And it was Schwartz's job to capture all of it. Every test, every sample, every strange result, through the lens of his camera. The cloth itself is simple to describe and impossible to explain. A linen sheet just over 14 feet long. A faint image of a man's body, front and back, as though the fabric had been laid lengthwise over a corpse, head at the center. At first glance, unremarkable. Sepia-toned, quiet. But the closer Schwartz looked, the less it behaved like anything he expected. There were no brushstrokes, no pigment, no layering of paint of any kind. Under magnification, the image seemed to exist only on the very outer surface of the linen fibers, a discoloration so thin it barely qualifies as a physical mark at all, with nothing beneath it. Dyes soak into fabric, paints soak into fabric. This did not. Then came the negative. When Schwartz developed his photographs and looked at them reversed, light for dark, dark for light, the image sharpened. Details that were murky and indistinct on the cloth itself resolved into something startlingly clear on the photographic negative, as if the shroud had been a negative all along, waiting more than 1,000 years for a technology that would not exist until the 1820s to finally reveal what it was actually showing. And there was a third detail, stranger than either of the first two. 2 years before Schwartz ever arrived in Turin, another team had run a photograph of the shroud through a NASA-developed device called a VP8 image analyzer, a machine built to turn a satellite photography into three-dimensional relief maps. Feed it an ordinary photograph or a painting, and it produces distorted nonsense because paint and light don't carry real distance information. Feed it the shroud, and it produced a coherent three-dimensional shape, a face, a body, as if the darkness and lightness of the image correlated not with how something looked, but with how far it had actually been from the body beneath the cloth. Schwortz watched this data confirmed again during the 1978 investigation. He has spent decades since explaining patiently to skeptics and believers alike that this is not something paintings do, ever. By 1981, when STURP finally published its official findings, the language was careful, almost cautious, considering what the team had spent years examining. The image, they concluded, was that of a real, scourged, crucified human form, not the product of an artist. The blood-like stains tested positive for hemoglobin and serum albumin, and then the report did something unusual for a scientific paper. It admitted defeat on the one question that mattered most. How was the image actually formed? Unknown. Unsolved. That word, unsolved, would follow Barrie Schwortz for the next 45 years. Then came 1988, a test that was supposed to end everything. The Vatican authorized radiocarbon dating, something STURP itself had never been allowed to do. Small samples, cut from one corner of the cloth, sent to 3 of the most respected laboratories in the world: Oxford, the University of Arizona, and the Swiss Federal Institute of Technology in Zurich. All 3 came back remarkably consistent. 13th to 14th century. Medieval. Centuries too late to have touched the body of anyone from the 1st century. For most of the world, the story ended there. Case closed. Forgery confirmed. But not for everyone who had actually studied the cloth up close, because the corner used for that test happened to sit near a section of the shroud with a documented history of repair and reweaving, the same general area most exposed to handling across centuries of public display. Some researchers, including scientists connected to the original STURP team, have argued in published work ever since that a single sample, taken from the most repaired, most handled section of a 2,000-year-old object, cannot reliably represent the age of the whole. Other scientists have defended the 1988 results just as firmly, pointing to the fact that 3 independent labs, working separately, arrived at strikingly similar numbers. That disagreement has never fully resolved. It is not settled science. It is a live scientific argument that has now run for nearly 4 decades. And it is the exact same tension between one confident measurement and the sheer physical complexity of an ancient, well-traveled textile that was about to resurface in a completely different form. Because carbon dating, it turned out, was never going to be the last word on the shroud's biology. In 2015, a separate team of Italian researchers examined dust particles collected from the cloth's surface and found traces of DNA, human DNA, but tangled together with plant material, hinting at a wide and unexpected geography. Genetic markers connected to populations across Europe and the Near East. A surprisingly strong signal tied to the Indian subcontinent. Intriguing, suggestive, but limited. The technology of 2015 could tell researchers roughly where a genetic trace seemed to originate. It could not tell them when that trace had arrived on the cloth, or separate one layer of contact from another with any real precision. For over a decade after that, the question sat there, unanswered, waiting for tools sensitive enough to actually crack it open. Then, in March of 2026, those tools arrived. A team of researchers led by geneticist Gianni Barcaccia of the University of Padua and Alessandro Achilli of the University of Pavia, working alongside forensic scientist Nomi Procopio of the University of Lancashire, published something that had never been attempted on this material before. Not a narrow test, hunting for one specific answer. A full metagenomic analysis, sequencing every single fragment of genetic material present in the sample, human and non-human alike, and matching each one against known genetic databases. The samples themselves were not new. They were linen filaments taken during that same legendary 1978 collection, preserved for decades by Italian forensic pathologist Pierluigi Baima Boglioni, waiting essentially for the science to catch up to them. Using a technique called PCR-free next-generation sequencing, a method built specifically to avoid the contamination and bias problems that plague older DNA testing, the team extracted and read genetic fragments from several strands taken directly from the body image area of the cloth. And what came back did not look like an answer. It looked like a mosaic, layer upon layer of biological information, none of it belonging to one single source. Several distinct human mitochondrial DNA lineages surfaced in the results. Genetic material passed down almost exclusively through the maternal line, mother to child, generation after generation. It cannot build a complete genetic identity. It cannot tell you what someone looked like. What it can do is anchor a person to a broad ancestral population, a maternal thread reaching backward through time. One lineage, called H2A2, matched the standard reference sequence used across human genetics research. Another, H1B, common throughout Western Eurasia. A third, H33, associated strongly with the Near East and notably frequent among the Druze population of the Levant. And then there was a fourth, K1A1B1A. This one, the researchers were able to trace with total confidence. It did not belong to some anonymous figure from antiquity. It matched the mitochondrial profile of the official collector who had gathered these very samples back in 1978. Sit with that for a moment. One of the clearest, most precisely identified genetic signatures recovered from the most scrutinized religious relic on Earth belonged not to a man from the first century, but to a scientist doing routine, careful, sterile collection work less than 50 years ago. It is not a failure of the study. It is, in its own way, the most important thing the study proved. It is confirmation that this technology works, that it can recover a genetic trace from this cloth and identify it with real precision. And in the same breath, it is proof of exactly how deep the contamination runs. If one 20th century handler left a footprint clear enough to be matched decades later, how many other footprints, from centuries of clergy, restorers, pilgrims, and researchers whose names were never recorded, are sitting in that same tangle, permanently unidentifiable? And the human DNA was only part of it. The samples also carried genetic material from cultivated plants, wheat, carrot, corn, even banana and peanut. From domesticated animals, cattle, pigs, dogs, cats, chickens, a dense community of skin-associated human microbes, fungi, including various molds, salt-tolerant microorganisms adapted to unusually dry, high salinity conditions, and, almost improbably, traces of Mediterranean red coral, a material long used in devotional objects across Catholic Europe. Two smaller threads, taken separately from the shroud's reliquary casing, were carbon-dated as well, and came back to 1534 and 1694. Both years fall squarely within documented periods of restoration and repair, including work carried out after a severe fire in 1532 nearly destroyed the cloth entirely. Piece by piece, a picture was forming. Not a frozen moment from 2,000 years ago. Something closer to sediment, centuries of hands, breath, storage, transport, and restoration all pressed into the same threads, with no boundary marking where one layer ends and the next begins. Here is where the story could have gone the way so many headlines wanted it to go. Proof of the impossible. Confirmation of the miraculous. It didn't. The 2026 findings do not identify the man in the image. They do not prove a supernatural origin. They do not overturn the 1988 dating, and they do not settle the debate around it either. What they reveal is something quieter, and in its own way far more haunting. Just how difficult, maybe how close to impossible, it may be to ever isolate one original signal from a cloth that has passed through an unknown, unknowable number of hands across an unbroken chain of centuries. Because DNA does not come with a date stamp. A genetic marker tracing back to the Near East could have arrived on that cloth in the first century, or it could have arrived with a single pilgrim passing through 600 years later. The new sequencing can tell researchers where a trace likely originated geographically. It still cannot tell them when. That is the wall this entire field has now run into. Not a lack of data, the opposite. There's more recoverable genetic material inside this cloth than anyone expected, even after nearly 2,000 years. The wall is time itself and the absence of any tool that can sort what's been found by when it arrived, rather than only where it came from. The public reaction split almost immediately, and predictably, along the same fault lines the Shroud has always produced. Some religious commentators pointed to the sheer biological complexity of the findings as evidence that the object only grows stranger the more closely it is examined. Scientists pushed back just as quickly, arguing that an old, frequently handled devotional object accumulating centuries of biological residue is exactly what anyone should expect to find. Nothing more mysterious than ordinary history, deposited one hand at a time. Neither side has definitively won that argument. Both readings of the same data are sitting in public right now, unresolved. And somewhere in the middle of all of it, once again, is Barrie Schwortz. Now in his late 70s, he has spent close to 5 decades maintaining Shroud. One of the most extensive public archives of Shroud research anywhere on the internet. A project he started in the mid-1990s and has kept running ever since, documenting every twist, every study, every disputed claim as it emerged. Across that long public record, his position has remained remarkably consistent. He has never claimed publicly that the Shroud's authenticity has been proven. What he has consistently argued, across decades of interviews and public writing, is that dismissing the object outright, ignoring its unexplained image formation, the unresolved questions around the 1988 sample, and now this tangled genetic record, is a position the evidence doesn't fully support either. A skeptical young photographer walked into that room in 1978, expecting to document a straightforward debunking. 48 years later, he is still there, still watching, still refusing to give either side the easy answer they want. So where does that leave the mystery after nearly half a century of investigation and a genetic study more sophisticated than anything the original STURP team could have imagined? In one sense, almost exactly where it started. The image remains unreplicated by any known technique, medieval or modern. The 1988 dating still stands, and the argument over whether it can be trusted still hasn't ended. But in another sense, everything has changed, because now there is proof, hard, sequenced, peer-reviewed proof that this cloth is not one clean biological artifact frozen at a single point in history. It is something closer to a palimpsest, a surface written over and over and over again, century after century, each new layer settling on top of the one before it, with no visible seam between them. The 2026 study did not solve the mystery of the Shroud of Turin. It did something almost more unsettling. It proved, for the first time, exactly how many layers that mystery actually has, and how much biological material may still be hiding inside those fibers, waiting for whatever tool comes after this one. Something that can finally do what this study could not. Sort the old from the recent, thread by thread, and find out whether the very first trace is still down there at all, or whether it was lost centuries ago, somewhere beneath everyone who touched this cloth and never left a name behind. If the genetic fingerprint of a careful, methodical man doing routine work in 1978 could still be read with total clarity 48 years later, a man who is, right now, still alive, still watching, still waiting for the next discovery the way he has waited for everyone before it, then what else is still down there, buried under centuries of hands that reached for the same cloth and vanished without a trace? Barrie Schwortz may never get his answer, but he is still looking. And as long as he is, so is the rest of us.